Thank you very much for joining us today. The topic today we're covering is how to manage your data with an electronic lab notebook. Really, we have invited two speakers today, Nathan and Markus, to kind of chat a little bit about how they got started with electronic lab notebook, what that transition looks like, and how they organize their data and files and inventory within the SciNote system. So before we get to the the speaker portion, I have a few housekeeping things. First of all, the webinar format, we're going to start with an introduction. I know that some people might not have seen SciNote in the past, so a very quick, super quick overview of the interface, and then we're going to go into the speaker presentations. At the end of the presentations, there will be a Q&A session. We have quite a few pre-submitted questions that we will answer. We will also answer the question you submit during the session, so please feel free to enter your questions into the Q&A box, and then we'll go from there. Also, make sure the webinar recording and resources will be available next week. So you will get an email in your inbox. So make sure, feel free to share that with anyone, your colleagues, or anyone who might be interested in learning about electronic lab notebook. And there's a post-webinar survey. We will share the link after the webinar session. Please make sure to fill that out. You'll also see that in a follow-up email. But yeah, that's an opportunity for you to share what you think of the webinar, or to let us know, if you have any questions about the webinar about SciNote. The next webinar on August 24th, leverage the API of an electronic lab notebook for efficient data management. We have two speakers, one from a contract research organization, the other one from a drug discovery company. They will be sharing how they use API to automate some parts of the data management. So yeah, if you look out for an email, that will be coming into your inbox. All right, that's all the housekeeping I have. I'm gonna quickly introduce our speakers. So we have two speakers joining us today. First of all, we have Dr. Nathan Tykocki. Dr. Tykocki's research focus is on urinary bladder physiology, with specific emphasis on the mechanism responsible for the sensation of bladder fullness. He employs a multidisciplinary approach, including biochemical, pharmacological and physiological and genetic tools to understand how bladder muscle, nerves, vasculature, and urothelium communicate to impact bladder function. Thank you for joining us, Nathan. And then we have another speaker, Dr. Markus Schosserer, has been the PI at the Medical University of Vienna Center for Pathobiochemistry and Genetics, Austria, since July 2022. He received his PhD in biotechnology from the University of Natural Resources and Life Sciences, Vienna, in 2012. His research interests lie in biogerontology, skin aging, cellular senescence, and RNA biology, particularly RNA modification, ribosomes, and protein translation. So SciNote, in case you haven't heard about us before, SciNote is a lab digitalization company with the mission to help humanity benefit the most from science and to preserve research data for future generations. Our electronic lab notebook is a leading ELN with inventory compliance and team management tools. We are currently the top-rated solution for the electronic lab notebooks category in G2, a third-party, one of the largest third-party review platform. SciNote is trusted by FDA, USDA, European Commission, and over 100,000 scientific professionals. And I also want to quickly mention, as you can see in the image there, that we recently launched our SciNote app for mobile devices. So you can actually take, SciNote into, on a tablet or a cell phone, and then into the bench, and then actually follow through your experiment using SciNote. So it's something that I want to quickly mention because we're really proud of that. So this is only a very quick mention also as well. We do have, so both Nathan and Markus are using more of our research lab kind of a plan, kind of a plan for a research lab at the moment. So we also offer a free version for personalized use only. So you obviously won't see like a team collaboration features within that personalized version. And there's only one inventory But you will see that the interface will, if you sign up for it, the interface is largely the same. So you will still see most of the things that Nathan and Markus covered during the talk in the free version, but you just miss quite a few features. So if you use the free version for a little bit, feel like you like it, make sure to get in touch with us to make sure you test all the features that's available within our premium version. Okay. Now I've done all the all the things I need to quickly mention about the company. Now I want to quickly mention the interface. I know that Nathan is going to go through that as well very quickly right after this, so I just want to briefly mention. So SciNote has when you sign to SciNote, it will look a little bit different from a few other platforms or electronic lab notebook or just the digital lab notebooks that you will see. You will first start to see a dashboard with a few different, with a calendar, with tasks that you need to be working on. If you have our feature that includes the signature option, then that will also show up in the dashboard. And then the SciNote structure is organized on the projects and experiments tasks level. So on the, we also have a, when you go to the project tab, you will see all the different projects that you organize within, SciNote. And then once you click on a project, within each project, you'll see the experiments that you're working on. And then once you click on the experiment, you will see the task. The task is actually can be viewed as a table or as a canvas. The canvas will show a visualized workflow. You can do linear steps or you can do non-linear steps, and that's extremely helpful when it comes to something you want to do some sort of parallel analysis of your testing sample. Or I think previously we've seen, a PI who do both behavior and, histological work for samples, and that kind of workflow allows allows you to branch out, allows you to organize your notes, not necessarily in a linear fashion, but in more of a, it can be simple or it can be complex way. And finally, you will also see once you click into a task, that will show you sort of like a protocol steps that you can check off. If you see this right away below, you will see kind of like a checkbox. But also on top, you get to include any details, notes. You can make references to other items within the electronic lab notebook. Finally, some additional things I want to mention is that we include a building inventory management system. I know that Markus is going to go more into this later on, so I won't talk too much about this, as well as a place for managing your protocol. So all the protocols are in one place, so you don't need to go to multiple places for all your protocols. Perfect. All right, I finished all my very quick download of SciNote. So I hope that's sort of like a very quick introduction. If you have any questions about SciNote after this event, feel free to, if you want to actually see in action, feel free to sign up for the free version or book a demo with us or sign up for the free trial. We're happy to do a demo also for a lab or institution. Just email us. Perfect. I think that's all. So I am going to hand this off to Nathan. Nathan, please go ahead. I will stop sharing so you can start.
All right. I hope I'm sharing properly. Well, good morning. Good. Well, good morning, everyone. Let me get my own picture out of my own way. So yeah, as Theresa said, I'm at Michigan State University. I'm assistant professor, and we work on lower urinary tract physiology, everything from in vivo work all the way down to ion channel work. And so SciNote has been, for us, kind of an invaluable tool for what we've been able to do and manage different people. So basically, in an overview of how we organize it, essentially we just started with grants as being our projects. So each individual grant is labeled accordingly, and we divide things within those grants into our different projects therein. And each of these sub-projects is in some cases broken down by type of experiment, by mouse strain, sometimes just depending on how it needs to go. We're able to use additional folders to track, you'll see some names there, those are undergraduate students that have been in the lab, to track different projects within these projects, because some of the things aren't necessarily exactly in line with the grant, but they're in line with different people. So it just allows the versatility for us to group things as they see fit into all these different sorts of stuff. And you can see at the bottom, what we do use pretty commonly is this ability to assign tasks and projects to different people. This has been something we've used extensively to be able to track people's work on a day-to-day, week-to-week, month-to-month basis, and also to know where to pick up when things are left off as, being in an academic setting, students that aren't graduate students are in constant flux, coming and going and coming and going. And that way we know how to keep them all together. From there, we use these project cards as, Valdo, this is my lab manager who took some of these pictures, set up to group these similar experiments together. So whenever we're doing all the zymography stuff, we have these cards that keep similar experiments together. I think for me, the real, and you can see under there, all of these different tasks that are associated with it. And this is really where the power of SciNote has come in for us, is this ability of how we decided to organize and group our protocols and experimental designs. So we use a lot of techniques in my lab, and we use a lot of them in multiple ways. So for example, isolated tissue bath studies or pressure myography. Parts of those experiments are key to every single day that we do. Parts of them are variable as the day goes by. And so this is just one way here that one of our new students decided to set up this is just a series of 4 days worth of experiments, where when building our protocols, we built the different steps associated with each portion of a tissue bath experiment as a different protocol. So setup was one, a concentration response curve is another, adding antagonists or agonists is another, and then cleaning up is another. And the reason we did this is because then we didn't have a bunch of steps in the in-between that we maybe didn't use or needed to tailor to something different every single time. We had the whole setup that we could just do, the setup the first time, and then different concentration response curves, electrical field stimulation, whatever is in between, and make these different things. So that addition, the reason that also ended up useful is that then if we're doing other experiments where we're adding, say, the same antagonist to an in vivo experiment or a pressure myography experiment, we don't again have to reinvent the wheel of the protocol. We just pop that in from the other from this other plan. And so this is one way that this was a brand new user set it up, but some of our more seasoned people set it up a little more like this. So when we're building an experiment, this was, you can see the top, it was for a colleague, some blinded experiments to stuff. On any given day, in this experiment, we received 3 bladders by FedEx. And so we were in a move to do them all. And so the making of buffer and the setup of the pressure myograph were the same with all of them. So we didn't need to repeat it, because once it was up, we just put a new vessel in. Now we have our 3 different blood vessels, 3 different experiments. And at the end, all of them led to being cleaned up. And so what was really great about this is the things we needed to track, we could track. The things we didn't need to track, we didn't need to repeat. And within all of these, we were able to go ahead, take notes, make comments throughout all of it, and track time of day and so on through all of it as the steps were completed, so long as our people go through and check it off. Pretty much all that we use without fail are the idea of these checklists and these protocols. This has been, for my lab, the greatest way to do it, because I know the date, the time, who it was, when it was done, and how it was completed. Sometimes you do have to go back and say, hey, why is this still open? And then people go back and fix it, but that's no different than a paper lab notebook. And wherever I am, I'm able to track this in time. And if somebody's having a problem, I can see exactly where the problem began in terms of what they did. And so it's really come to be extremely useful in just the setup, the design, and management of my people and the projects that we're doing and the experiments that we're doing and when we're doing it. And so I don't want to blab on for too terribly far ever. And so what we don't use a lot of are the inventory management, because we don't have a lot of samples and cells lines or anything like that in order to manage. But I think that's way more in Markus's wheelhouse. So Markus, I'm going to stop sharing here and allow you to now take over and talk about how you use this. All right.
Perfect. While we're in the transition, Nathan, can you share a little bit as to How did you get started with the electronic lab notebook? Like, why did you decide to switch from paper to ELN, and how did you end up with SciNote actually?
Yeah, so we decided to switch. When I started my lab, I come from a lab of just me at the University of Vermont. After I was a postdoc, I was a research assistant professor, and it was all paper. And then I realized when moving, that became really complicated. Of what data go where, being able to track these things. And also, I'm really, really poor at keeping a good written lab notebook and always have been. I hate to admit that, but it's true. And so this was a way that kind of fit within my tech abilities, because I like techie things, but also centralized everyone in the lab to be able to do that. And, all of our grants are National Institutes of Health funded, and we have requirements for maintaining data and data integrity over time. And with this being, servers offsite, all of that, at the same time where we started this, I had a friend whose lab completely flooded from a broken water pipe, doing about $10 million worth of damage to equipment and stuff. And at that point, I was like, no, we cannot have this on paper. We can't have something that can drown in a puddle, or that I'm going to pour coffee all over, or is going to get lost in the shuffle of students in and out. We need something that's centralized, and also that tracks everything. So that's really why we got started on it.
Right.
And we tried the other ones too, but the visual interface of SciNote to us was hands down the thing that won. The little blocks and all of that. I'm a very visual person, and this really made sense with my brain.
Right. That's great. Thank you for sharing. I have the same experience when I was in school. Not my lab, but the next door lab. Got burned down, and all the lab notes were lost. And I was one of the terrible undergrad students who took the lab notes home. I know my PI at that point probably wouldn't be too happy about that. But I got lucky because of that. I had my undergrad thesis that I could still write up. But yeah, totally get it. If there is a fire or flood or even simple student turnover, not being able to track or the potential of losing those notes were pretty tremendous, and it's kind of terrifying. Markus, sorry about using a little bit of the time. Please go ahead with your portion.
Thanks a lot, and hello, good afternoon from Vienna. So as Theresa already introduced, I am independent PI since July 2022. And in my group, beside myself, there we have 2 postdocs, 2 PhD students, 1 lab manager, and we always usually have 2 undergraduate students or interns, which stay for around 3 to 4 or 5 months maybe. So in total, we are a group of around 6 to 7, 8 people. My research is focused on biological aging research, and we are studying when we use different models systems for studying aging. So on the one hand, we're using cellular senescence of primary human, mostly skin cells, but also other cell types. Then from those cells, we also make 3D organoid models, for instance, skin models. But we also want to understand aging processes at the organism level. And for this, for instance, we use Caenorhabditis elegans, so nematode worms, and also we do some experiments in mice. The specific aspect of aging we are studying are RNA modifications. So at the moment, we are mapping different types of RNA modifications by direct RNA nanopore sequencing. And we would like to understand how these RNA modifications impact aging, but on a mechanistic level, especially also how they influence mRNA translation, cell behavior, and also the physiology of an organism. The idea to use an ELN already came up during my time before I got my independent group leader position. So from 2012 to 2022, I was postdoc and also co-PI at a different university in Vienna. And there we used many different things of documenting our data. So we, of course, use these conventional paper lab notebooks for each team member. This then for PhD students accumulated to 8 to 9 paper notebooks during their scientific career. We then also, so we started with some Word-based protocols, but this then became very large, and then we decided to use a wiki-based protocol collection where we can also add some search functions. Then we use Quartzi for ordering and some sort of general inventory for general chemicals. We had many different Excel sheets as inventory for primers, plasmids, inventories of different PhD students, and so on and so on. And then we also had a specific online database for the whole institute, comprising cell banks, liquid nitrogen. So many different data sources. And this I found very challenging because for getting some specific information, you had to rely on many different data sources to get all the information together. And another big challenge was, especially in the last few years, and especially during the Corona pandemic, was finding all research data in paper notebooks, especially from undergraduate students, which left maybe 2 years ago, and then finding some specific data in their lab notebooks was very tedious and took a lot of time. And especially also the traceability of reagents. So what do I mean by that? So for instance, in a lab notebook, you find information that a student used an antibody against mTOR, for instance, but we have 3 different antibodies against mTOR in our lab over the years. And then there's the big question, okay, which antibody was it that was specifically used? So during those 10 years as postdoc and co-PI, I became very motivated to establish an ELN at some point when I became an independent group leader. And this is then actually what we did when I moved to the Medical University last July. How did we decide on SciNote? So we decided to manage it like this. We tested 4 different ELN solutions, both commercial and free. And right from the beginning, I involved my team members. So I didn't want to do a top-down solution so that I say, well, okay, we use this solution and everyone has adapted to that. I really wanted also to have some feedback from my team members. So we tested 4 different solutions. We immediately saw that there are large differences in the user interface and the functionality. And therefore, of course, these ELNs are highly personal. So some people will have a certain preference for one solution or for another solution, just depending on the individual working style. And what we also saw that some ELNs are highly polarizing. So some ELN solutions, some people really loved and some people completely hated and didn't like at all. Again, depending a lot on the individual working style. And what we also saw that some ELNs are specifically tailored for specific means. So for instance, there were some ELNs which work very good for chemists maybe, and some of them work very well for molecular biologists. But since we are doing quite a bunch of different methods, we didn't also want to rely on a very specialized solution, which was not really adaptable to our needs. And therefore, then in the end, we decided for SciNote. It was my favorite ELN, especially because I'm a very visual guy and I like this very clear structure with project, experiments, tasks, which Nathan already introduced you to, and also this graphical representation of flowcharts I really liked. And it was also a solution which all lab members really liked and could live with. And therefore, in the end, we decided then for SciNote. And what I especially also like about SciNote is that you can in principle link everything with everything. So it's one solution where you can have your lab notebook, but you can also have your inventory lists and your protocol steps, and you can link everything with everything, making the traceability much easier. Okay, so for setting up SciNote in our lab, so as I told you, we had our information distributed over many different channels. So the protocols from Wiki and Word files, this was maybe the most tedious part we had to do because we just had to simply copy and paste everything into SciNote to set up the protocols. What we figured out during the process definitely pays off to really take a little bit more time and really copy every individual protocol step to get it into the structure of SciNotes with this individual protocol steps, because only this structure really allows you then in the end, if you use it at the bench with your mobile device or your mobile phone or your tablet, because then you can really check each individual item and say, okay, it's done now. If you just copy the whole protocol as it is like this into a single task, then it's more complicated because then you lose this functionality really to tick off the individual items. And another thing you have to be aware of is that protocols at the moment can only be modified by the creator of the protocol. So probably if a student sets up a protocol, it may be a good idea before you import it into SciNote just to check it for any logical errors, typos, and so on. Because in the end, it's super annoying if there's a protocol in there with a lot of typos and errors, and then only the person actually who set up the protocol can edit it in the end. And then for migrating the inventories from different sources, this was much easier. We already had some inventories already in Excel files or CSV files. And for instance, from Quatsy, you can also very easily export all your Quatsy data into a CSV file. The only restriction is the individual items should be organized in lines, and the categories for your inventory should be organized in columns. How you then actually call the columns in the Excel sheet doesn't really matter, because this you can anyway then specify in SciNote. So for instance, in SciNote, if you import an Excel sheet into SciNote, SciNote tells you, yeah, well, you have, for instance, the storage location, which column in your Excel sheet corresponds to the storage location. And also another hint, which I found very important, is to carefully plan the structure of your of a new SciNote inventory, especially how many different categories you want to have, how you name the categories, and especially also regarding the data types before you do the import, because some things are difficult to change afterwards, especially the data type in the category. What I mean by that, I will show you in a second when I go through how we set up the inventories in our lab. So at the moment we have 5 different inventories, and I want to briefly go with you over the different categories just to show you how we organize the data. So we have one general category where we store all of our general reagents which we order and which we use. So you see all the different things there. We have a category for specifying the manufacturer, the product number. When did we add it, who added it, then the location, sub-location, and the dates and so on. And now just to come back to the data category in the different columns. So for instance, for the product number, it is not a good idea for using the number category for the product number, because you see some product numbers do not only consist of real numbers, but also of letters like T and ML. And if you just define this as a number, then you would lose this information. Another thing is, for instance, for the location, we use just a drop-down menu where if you have, for instance, 20 or 30 different locations where you can store the item, these drop-down menus are really nice, because then you can just select where you stored the reagent. And it's also less error prone, but we also have a sub-location, and this is just a free text box where we can just enter the sub-locations. Because sub-location, this would be a list of 500 items, and then finding something is really, really tedious. And then what I also like is to have in almost all inventories we use, we have just a general free text section, comments, where you can just put any information which you like and what you want to share with the other users. So for instance, in this case, we added some information that the reagent is toxic. Or here you see we also included some hyperlinks to the internet where you can find the protocol of how to use the specific reagent. Then we have one category where we organized our -80 inventory. In this case, we have some general boxes where we just store reagents which are for general use, like for instance lentiviral particles, but we also have individual boxes for each team members where they store the individual samples. So for us, it would be too tedious to make a single inventory item for every single sample which every lab member generates, but instead we make designate a box for each team member or 2 boxes, and then the team members have to add information what is actually included in these boxes. So for instance, RNA isolation samples from this and that date, and so on and so on. And of course, also the rack number and the box number and the storage location in the -80. Something similar we did for the cell storage in the liquid nitrogen. Again, there we have the first few columns define where the reagent is stored. Then we have again some free text commands. So for instance, the cells don't grow that well after you thaw them, things like that. And then we also found it very useful to add a category about handling. And there we have hyperlinks, as you can see, with the reagent category. So for instance, here we have a specific medium which we use for passaging those cells. And if I would now click here on RPMI 1640, then the information box would drop out, linking this item with the other inventory and telling me exactly where I find this item and how to use it, and also some other information. Then we have one inventory for our plasmids. There, for instance, we add information about plasmid backbone, insert, where we store the bacterial stock, which bacteria strain is it. If we have a DNA stock at -20 degrees, where do we store it? And again, some free commands. Yeah, and something similar, of course, you can also do with primer categories, antibodies, or whatever you like. And then last but not least, I also would like to briefly talk about how we organize our research data. So similar to Nathan, we organize the data in projects, but in our case, each project reflects roughly a planned manuscript. So we, which of course also partially overlaps then with grants which we get. But we also have general projects. So we have a general project for cell culture passaging, which I will show you in a moment. And this project we archive after each quarter. So you also want to think about regularly archiving your project, because else you will end up with 50, 60 different projects, and then it becomes too much and not very nicely viewable anymore. And then we also have general projects when a new team member joins our group for onboarding, and also manuals for different instruments. In a hindsight, this maybe would fit better to a protocol. So instead of making a project with that, you could also make a protocol with that. Yeah, and as what I mentioned before, is I find it very, very useful, and I can highly recommend to use this option, is linking inventory items to task protocol templates, because with this you solve this problem about traceability, and this is super convenient. So for instance, if you do if you run a protocol, in the protocol you see, okay, I have to use this specific prime or this specific probe. You just click on it, and then this nice information box pops up, where you immediately see the storage location where you can actually find this, and also some other useful information. This is, for instance, how we organize our cell maintenance experiments. So we usually do it. So we usually keep it only. So we have one specific experiment for each cell line which we are passaging, and we do it in a quarterly manner. And then if we zoom in, so for instance, we see HeLa cells we passaged twice per week. And sometimes you forget to really mark a task as completed, as you can see here. And to avoid this, we also just add somewhere else the information when this task was actually really done. So sometimes the information that the task was completed, like here, doesn't really fit with the time when the task was really performed, but you have a lot of different options in SciNote just to add this somewhere in the notes. Okay, with this, I'm at the end of my presentation, and I would be happy to discuss further and also take your questions.
Thank you so much, Markus. Perfect. So now we are at the Q&A session, and I see a lot of questions being asked. So a few things. Laura, my colleague Laura, is going to put the post-event survey into the chat box. So after the event, please make sure to fill out the survey. Okay, we're going to start with the questions. Thank you, Markus, for sharing all that. Okay, good. So we have a few things. I think we have a few pre-submitted questions, and we have a few live questions. Any questions that we don't answer during the Q&A, we'll try to see if there are other ways that we can cover them in a blog post or follow-up email. So I think one question that came up quite a bit is on the use of a SciNote in the lab. So for day-to-day at the, either at the bench or in the lab, like how are people using it? Like, do you have multiple members? Like, do you have just a few people using it? Do you have the whole lab using it? Do they use it? I'm sorry, combining a few questions into this one. Oh, sorry. I also just remember, before we go into the Q&A session, our colleague Arun, who is a customer success manager, is now joining us, also to kind of help to see if there are questions that he can help answer. Sorry, Arun, I totally forgot to introduce you. Perfect. All right. So the first question is, how is SciNote being used in a day-to-day environment in your lab? For example, are people using it at the bench? Are there only specific people using it, or all the team members use it in the lab? Nathan, maybe I'll have you answer that to start, and then we can go to Markus.
Yeah, for us, everybody uses it. We don't have a choice, because if I gave choices, things end up all over the place. So yeah. The only place it gets weird is with undergrads because of the constant flux of people moving. So we have a dedicated account that is undergrads, because it may be the same person, one person 1 week and one person the next. I know SciNote might not like to hear that, but that one undergrad computer has its own thing for those. But everyone else in the lab has their own. My lab manager, each graduate student, both technicians, everyone has their own, and everyone is expected to put all of their stuff in it and follow it.
Markus, how about you?
It's exactly the same for us. So also everyone in the lab is using it. In the data, we also have it, like Nathan said, that we have one account for all undergraduate students just because they change so much, but for all regular members of the, for all members of the group which stay for longer, they have their own SciNote account. Usually people use it like that they still take that they still have a paper notebook where they just take notes while they are working in the lab, and they usually also have the mobile phone or a tablet where they can look at SciNote at the protocol just to really kick off the steps, and then afterwards when they are back in the office or at the computer, they really nicely then protocol everything in SciNote. But I guess SciNote is working already on that in the future it will also be more integrated, that you actually also can and will take notes also in the mobile app.
Perfect. That sounds great. Excellent. Arun, do you have anything to add? Like, what do you see users doing day-to-day kind of data management on your side as a customer success manager who helps everyone kind of doing their onboarding and training?
So from my end, I would say our more successful users of SciNote tend to have a a top-down mandate that everyone needs to use SciNote. Like Nathan said, if it's left optional, then you always have people choosing to use Google Drive or SharePoint or just keep it on their computer locally. So I think that really helps. With regards to a constant turnover of undergrad students or visiting researchers, I've seen both approaches where they just have a dedicated account called for the undergrads, or they do have an account for the incoming student. And when that student leaves, you can remove them from SciNote. And when you remove them, the ownership of their data gets transferred to whoever the team owner is. But the advantage of doing it this way is you still retain who worked on that data in your historical records. So if you need to go back and revisit a result or validate something, you can always see which undergrad student it was because it'll have their name on there. And even though that license is now being reused by someone else.
Perfect. Oops, sorry. I accidentally moved my slide. Thank you so much. All right. Thank you all for that, answering that question. We have the next question I want to answer is about Quartzy. Quartzy is being used widely by everybody, I believe. So the question is, more specifically Markus, but I know that Nathan also uses Quartzy because we talk about this in our rehearsal. How do you manage ordering after moving to Quartzy? Did you use API or another order management system, or does it generate a reorder list that you can take and search vendor catalog?
I have to admit that at the new university where I am, so at the old university was handled that the ordering was done for a rather large group and for several research groups together. Therefore, Quartzy was really convenient. Now at my new university, we're doing the ordering just for our group, and that's actually sufficient for us to use Excel sheets. So we did not, so we are not using Quartzy anymore just for ordering, we're using Excel sheets. But then as soon as the items arrived, it is mandatory to put them into a SciNote inventory, and this works really well.
So Nathan, do you have anything to add for how you handle that?
Yeah, for us right now, they're handled completely separately. our physical inventory in the whole lab and then the use of SciNote are independent. So that idea of connecting the two at some point in the future, that is super. We haven't run into any issues with having it as such, but yeah, they're just two separate entities to us.
Yeah, and I think I know that there's been Quartzy has been actually sharing this information on their channel, and we're actually exploring some sort of integration at the moment. So I'm hoping, crossing my fingers, that's going to happen fairly soon, but I don't have a timeline yet. So we're exploring a integration where you can kind of see the Quartzy inventory within SciNote, and then kind of jump into Quartzy for ordering. So that's something that we're exploring. And then if there's also obviously the option for API. So I want to touch briefly on this because we have a lot of questions. So Arun, can you chat a little bit about, just slightly about, quickly about our API?
Yeah, absolutely. So our API is freely available to all of our premium SciNote users. Our API documentation's on GitHub. It gives you all the information you need to know with regards to what API endpoints SciNote has. And each API endpoint basically dictates what data point in SciNote can be accessed via API. This includes things like being able to add new inventory items, update existing items, fetch attachments, results, steps, all of these things. And you'll find the entire list on our GitHub. And if this is something you are interested in, you can always reach out to your CSM, myself, or just our support email, and we can help you with that. But I've seen, from my experience, I've seen our users use API with a few different use cases. One tends to be integrating with an external database, whether it's an inventory system, another file management system, or something else, or to connect to some of their lab equipment. They want to export the data when a run is complete directly into SciNote. So this requires the corresponding lab equipment or software to also have an open API with endpoints that are accessible. So once it has that, you can get it to talk to SciNote with a little bit of scripting or programming on your end.
Thank you, Arun. And I know that definitely there are users who use API to help manage inventory. I know our one of the users, Wagner Institute, also does it that way. So that's their information is available on our website. So if anyone's interested in how they use it, we also have a API webinar on the 24th of August. So make sure to join us if you're interested in more in the API. So I have a next question is about transition. So there are some questions about how students found the user experience easy to adopt or transition or to learn. There have also been some questions about, how do you introduce the ELN for projects that are already ongoing? So that transition, from other platforms or from previous experiences using paper notebook to now using the ELN. I'm combining two questions here, so feel free to let me know if you want to split it up. But what kind of experience was that like? Markus, do you want to start?
Yes. So in our case, of course, it's a challenge for ongoing projects to move from paper notebooks to Quartzy. To SciNote. But it also works. We just because at some point you just have to start. So we just started using SciNote, and then and just for this specific project, then which is ongoing, we have it. We have the information double. So on the one hand, we have it in the paper notebook, and on the other hand, we have the new information in SciNote. It's a little bit tedious, but at some point, of course, you have to start with adapting it. Regarding experience of students, so I found it especially undergraduate, graduate students who have not a lot of experience. They really love SciNote. They immediately jump into it. They're highly motivated, and they're also really proficient using it pretty soon. People who are used a lot, who already developed their own working style with paper notebooks, they are sometimes a little bit more hesitant to move to SciNote, and they usually need a little bit more pushing and motivation, really, to let their paper notebooks go and then use SciNote. But for people who are completely new, for them it's actually easier than for others, in my experience.
Yeah, thank you, Markus. And I know that you also use SciNote during your lab meetings, right? To make sure that everyone kind of get into that kind of using SciNote experience kind
exactly,
of thing.
Yes.
Yeah. Perfect. Nathan, do you have anything to add from your experience?
No, it was about the exact same. We just kind of ripped the Band-Aid off and started using it, and it just numbered all the old lab notebooks, and at the top of experiments that crossed over said the rest of this can be found in lab notebook 71 pages 18 to 22 or whatever it is. And it's no harder to learn than it is to teach a new user how to set up a lab notebook properly. So it's the same thing. So it's not been a problem at all.
Good point. Arun, do you have anything to add based on your experience of helping users?
So from my experience, it's been very similar to what Markus and Nathan mentioned. There's always the initial inertia to adapt a new software, especially if you're used to working with paper notebooks. But once you overcome the initial inertia, it's pretty much a lot easier from there onwards. And with the legacy data or historical data, there are ways of bringing it into SciNote. I have some users that just create a dedicated team in SciNote and just label it legacy data, and slowly over time scan in their documents or drop their files in there that are on Box or Google Drive. And there is a way to automate this as well. Once again, with API, I had a user that did that. So it's the initial inertia that usually ends up being the roadblock. But once you overcome that, over time, everyone gets used to SciNote, and it's a lot easier with everything managing your data in a central platform.
Thank you, Arun. And I think between the two of us also chatted about this. I think it's just that at some point you just have to start using it because then otherwise it will never happen. So I personally, I like the Nathan approach of just rip it just has to happen. Okay, so we have some more questions coming in. The question is, if I need to see my student uploaded data and after, do I need to lock it down so no one can replace it or change it further? Do you have any policy when it comes to what to save, whether to lock things down or whether things need to be cleaned up, something like that when it comes to the data that you save? Nathan, do you want to start?
Yeah, so we generate an obscene amount of data, imaging data in my lab. We do lots of crazy imaging stuff and, terabytes of data a month that go well beyond what we're willing to store in SciNote. And so when it comes to the data stuff, we have other cloud-based server-controlled stuff from our university that stores all of it. And so what's expected, that I expect of my students, is that I always know exactly where, in SciNote, where the original data for that experiment can be found. If they want to upload different things and different iterations of stuff of how they've analyzed it or reanalyzed it, okay, so we can talk about that later. But for me as a PI, the most important thing to me is that link between that experiment and the raw data that did it. Some things that are small, the raw data just go with it, like, a lab chart file from concentration response curves or things like that. But that's the bit of things that I audit, I guess, for lack of a better word, and that with the sort of things that are, so to say, locked down. After that, it's okay to me that things can be a little more fluid, but you always have a history of everything that happened to any experiment or anything at all in SciNote, so even nothing can get changed in a vacuum. You can't get anything changed without being able to see that it was changed. So that's
true, because there is an activity log that you can view to see what changes made, and that's why ELN can be such a powerful tool, because you don't necessarily get that information when you have to go back to paper or other platforms.
Yeah, you can't tear out a page or anything like that. It's all there always.
Yes. Markus, do you have anything to add, or do you use a similar process?
For us, it's very, very similar. So usually data which is not too large, we upload the raw data, for instance, Excel sheets where we do some mouse physiology things and so on. This I always ask my students to upload the raw data to SciNote together with the process data. And for more data intensive experiments like imaging experiments, RNA-seq and so on, everyone needs to provide a link where the original data and the raw data is stored. But I also always ask them to also upload some sort of summary process data or whatever. So just that after 5 years, when I look in this experiment, that I know, okay, what was the outcome of this specific experiment? This is something I want to have. It can be a chart, process data, whatever, but I just want to know, okay, what came out, what was the result?
Perfect. Thank you so much. So I think I have a question about linking, data into SciNote to other, external file storage sources. Do you have any thoughts on how do you protect those links from changing directory paths? I think that's something that's fairly hard to control within SciNote, but if you two have like system in place, you know, to kind of you know, make sure those links are like stay in place, like, you know, do you have suggestions there? No.
No, I mean, that's
Yeah, this feels
beyond
like
the scope
something
of what
that's
SciNote. Yeah, that's I mean, the sooner or later I can ruin everything. I mean, we've for a long time used conventions about how, like, exactly how dates are to be listed in everything. Like, it's the exact same way all the time. And, you know, we've talked about every file is going to have this, and this has really been part of NIH's new data management policies that they require to. But yeah, no, there's no good way. That's just how we do it, but there's no way to force it.
Yeah, exactly the same for us.
Perfect. All right, so I have one question about students, because as you know, PIs working in academic institutions, they're always influx and, you know, students. How do you make sure students actually put all their work in SciNote? Markus, do you want to go ahead with that?
Of course, I can't really make sure, so I'm not really the person who checks every steps of every student. But we have our weekly lab meetings, and we sometimes or often use SciNote. Actually, during the lab meetings, we just project the SciNote where we look at the different projects and the data. And by this, on the one hand, I understand what the students did, and on the other hand, if it's really documented in SciNote. And this is then also a good motivation then really to keep SciNote up to date if everyone sees it, and yeah.
Yep, same. I just look at things on Friday morning and make sure that something from the week was there.
Okay, perfect. Thank you so much. All right. Okay, good. Sorry, I'm looking at all the questions. I'm checking like the Q&A and the survey and our pre-submitted questions. So I want to make sure. So I have a question here that whether there are tools for processing and visualizing raw data. I don't think those tools are in SciNote. Really, SciNote serves as kind of like a center for kind of, you know, data management almost in a way. So usually those tools for processing and visualization of raw data are done off SciNote, and then either you can provide a link to those data or that information. I know that, Arun, do you have anything to add here? What have you seen other teams doing?
Yeah, so generally, when files are a proprietary format, you can't really open it in SciNote. So you can store them in SciNote. You can store any file type in SciNote. But if you need to work on it, you'll have to do it outside of SciNote and then re-upload the analyzed data or the edited file or updated file to SciNote. Right. But the nice thing is the activity log or audit trail will tell you when a file has been uploaded or has been modified or anything like that. So that history is there, but unfortunately we can't support all data formats to be opened within SciNote.
You can open and edit Word document, you know, Excel, and can you elaborate on our Microsoft integrations actually? Yes,
so we have an Office Online integration. So you can open Excel files, Word documents, or PowerPoint documents right within SciNote, make all the edits within SciNote, and the changes are auto-saved. This is usually used a lot by teams that have multiple people collaborating. So if I start off working on an Excel file, doing my data analysis, someone else can pick up where I left off tomorrow, and they can just find that Excel file right in SciNote in the dedicated project or experiment.
Perfect. Okay, that's great to know. I also want to mention that we are also looking at integration with a platform for Ganymede, which is an online virtual platform that allows easy communication with, like, you know, other virtual, you know, other analysis software. Those are this kind of integration is more for larger, you know, drug discovery companies from what I've seen, but I don't know what that will come out. So, you know, we'll see if there's potential for that as well. Okay, so let me see if there are other questions. I think, sorry. All right, I think, is there any questions that we haven't answered? You know, anything else you two want to add to kind of like how you've been using SciNote and the benefits you've seen with using SciNote for data management? Oh, sorry, I should specify. Nathan, do you want to go ahead?
No, I really have nothing else to add. I mean, it's just the versatility has proved great. It's been less of a hassle than the paper lab notebooks for me, because I mean, that's the key thing. Like, I can be anywhere giving a talk or something and have a question about raw data, you know, when I'm making slides on a plane, you know, I can go back and pull it at an instant. It's been fantastic. I mean, even, you know, from your phone at a meeting. And so just having all of that with you all the time is indispensable.
Markus, do you have anything to add?
Similar, so I also see mostly benefits. Most of my team members, after some time, really adapt to it and really like it, and also use it a lot. For me, of course, it was a little bit easier to adapt it to our working style, because I changed university, I set up my new lab, and of course, if you're in a situation like this and setting up a new lab, then completely changing from lab notebooks to an ELN is very easy. In a running lab, it might be a little bit more tricky, but it's definitely worth the effort, I think.
Right. Yeah, and I think the SciNote, I think it's also I think people can also see that, like, you two have a bit different structure of how you manage projects and flowcharts, and I want to mention also that, like, things like projects, experiments, and tasks is more SciNote terminology, but really you can have, you know, a grant as a project. I've seen animal models as project. I've seen cell lines as project. I've seen all kinds of stuff as project. So it's really a hierarchy providing for people to kind of organize their information, really. All right. So if you have any questions, please free to, again, please fill out the survey. If you have any questions, please contact us through the email. You know, our team is around to help out, and let us know if you need to take that first step. We're happy to help. You can see our website for more information. Yeah, so I think that's everything. Thank you all for joining us. Thank you, Nathan and Markus. Thank you, Arun, as well, for answering some of the questions. We'll see you at the next webinar. Bye.